So why talk about all those different types of RNA? Because we're going to go back to our central dogma, DNA, RNA, protein. And we're going to use all those types of RNA. So having a good idea what they do will help us here. Let's talk about how we get from DNA to RNA. The first thing we have to do is go through a process called transcription. It's the process of reading DNA and changing it to mRNA. You're going to notice some similarities to DNA replication, but of course it's different because it has a different function. In eukaryotic cells, this is going to happen within the nucleus. That's where your DNA is. Transcription will happen there. You'll make your mRNA. It will leave through your nuclear pores, go to your ribosomes. In prokaryotic cells, remember your DNA is circular and it's not enclosed in a nucleus. We say it's naked DNA. So it's going to happen in the cytoplasm, but it will still move on to your ribosomes to go through a process called translation. This slide really just tells you all the different words that we're going to use and vocabulary. You should kind of become fluent in them, right? You should be able to use them, explain them, and know what they mean freely and easily by the end of this. So let's dive right into the process of transcription. Okay, we're going to have our transcribed DNA strand. This is also known as our template strand, our non-coding strand, our minus strand, or our antisense strand. All of those mean the same. In our diagram here is my template strand. Then I have an untranscribed DNA strand. It's called my coding strand. And we call it my coding strand because it contains the gene that we're going to basically use as our mRNA sequence. It's really the gene that we are coding for. Why is that? Well, because my mRNA sequence will match this coding strand nearly identically, right? Except for instead of thymine, I'm going to have uracil. So they're the same. It codes for the mRNA strand that we use, but we have to use the template strand to make that mRNA strand. We have this happen in what we call a transcription bubble, and we have to use a very specific enzyme called RNA polymerase. It's going to start at a place called a promoter and stop at your termination sequence. Then your mRNA strand will leave through your nuclear pore. Now, just as RNA had direction, so does just as DNA had direction, so does RNA. So we're going to build our RNA strand from the 5' end to the 3' end, meaning we're going to add one nucleotide at a time to our matching strand until we get to our termination sequence. We're going to add to the 3' end of our RNA molecule. That means that we're reading the DNA strand in the opposite direction, right? If we build 5' to 3', that means we're reading 3' to 5' on our DNA strand. RNA polymerase will add these RNA molecules one at a time, just as we saw in DNA replication with DNA polymerase. And then when it's done, this mRNA sequence will release. And if it's a eukaryotic cell, we'll go through some modifications, but it's pretty much done there. So here's another picture of transcription. You could use this picture to kind of walk yourself through the process. Make sure you can identify the RNA. You can see that here are RNA bases, our RNA nucleotides. They're going to be in the nucleus kind of ready for us to use and add on to our mRNA strand. Remember, mRNA stands for messenger RNA. It's going to carry the message to the other parts of the cell for us. So now might be a good time to go ahead and pause this, walk yourself through it, make sure you understand everything. I know we already talked about this, but I want to make sure you have it written down and you know. Remember how DNA had direction, we could only add to the 3' end, right? RNA is the same way. We add our nucleotides to the 3' end. That means RNA polymerase is reading DNA from the 3' end to the 5' end. But we're building our RNA from the 5' end to the 3' end. And you can see that actually in kind of 2 out of 3 types of the RNA we talked about. Our mRNA, we'll see it in our tRNA, although we don't reference it as much. And it would be in our ribosomal RNA too, but we don't have to worry about it so much. So here's our T terminus and our 3' terminus, and that's important to know. You should also be really good at transcribing and translating or changing your code from one form to another. So I want to give you a second. Let's say this is your coding DNA strand. Can you figure out what your template strands code would be? And can you figure out what your mRNA code would be? We'll go ahead and do tRNA and amino acid sequence later on. Take a moment, pause, and then I'll show you the answer. Okay, here's your answer. What do you notice? Well, you should notice that your coding strand and your mRNA strand are exactly the same. So what's important here? Well, mRNA, we're going to say, carries our codons. They're going to code for what amino acid we need to add to our polypeptide chain, which comes from our coding strand. And amino acids are going to be coded for by 3 nucleotides. So UAG is going to code for a specific start codon. We call it CCC would code for an amino acid. GCU would code for an amino acid. CGA would code for an amino acid. And remember, we have 20 amino acids, so we're going to use lots of different codes to sometimes even code for the same amino acid. But you should be comfortable going from DNA to DNA to mRNA in any order or form. When we start talking about transcription, we'll do tRNA and our amino acid sequence as well and talk about how we use that.